Hello, so I will be talking about the distribution of the lancelet Branchiostoma caribaeum, which is found in the phylum Chordata, subphylum Cephalochordata, and how it's affected by sewage discharge. Um, sorry. The biomass and the density of Branchiostoma caribaeum was analyzed along with the sewage by fecal steroids in a subtropical estuary within southern Brazil, known as Paranagua Bay, and the Caatinga Channel in Paranagua Bay. This experiment highlights usefulness of a multi-scale perspective to evaluate distribution patterns of benthic invertebrates as an indicator of environmental pollution and eutrophication. So, benthic organisms are used to detect the human impact on the environment as they reflect the environmental conditions that are studied. Benthic organisms are indicators of anthropogenic transformations due to their responses to exposure stress. Increasing eutrophication by sewage contamination is among the main causes of the degradation of water quality within these estuaries, and the analysis of the benthic responses to eutrophication have intensified along like the southwestern Atlantic coastal regions. Eutrophication is the excessive richness of nutrients within water due to the runoff from land that causes growth of plants and lack of oxygen. Paranagua Bay is one of the largest, most preserved estuaries in the southern Atlantic and houses one of the largest ports within the country with about 140,000 inhabitants. High concentrations of trace metals and sewage contaminations were recorded within Paranagua Bay, and the only species of lancelet reportedly found within the Brazilian waters was Branchiostoma caribaeum. And generally, lancelets are associated with oxygenated coarse sands, and density normally decreases in areas under environmental disturbance, such as organic pollution or eutrophication. A nested sampling approach allowed for a more complete investigation regarding possible environmental factors that can influence spatial variability of Branchiostoma caribaeum. There was two hypotheses tested. One, the largest source of spatial variability is expected at the sector scale, and two, density and biomass of Branchiostoma caribaeum are expected to be lower near the source of domestic influences. The differences within the lancelet species share the same feeding strategies and are very common in coastal sediments worldwide. This study evaluates the patterns of variability within the density and biomass of these specific lancelets at 4 spatial stages along a gradient of sewage contamination within the Caatinga Channel in Paranagua Bay, defined by the concentration of fecal steroids. So there are 7 materials and methods that they use, but the 3 that are most important are the study area, sample design, and field percentages and data analysis. So the study area obviously is the Paranagua Bay, one of the largest estuarine systems within South America, and it includes the Caatinga Channel, which is mainly focused on, and sampling design and field percentages. So the Caatinga Channel is subdivided into 3 sectors, the inner, middle, and outer. 3 areas separated by hundreds of meters were randomly established within these 3 sectors, and 3 sites separated by up to 10 meters and 5 replicates, each spaced apart at the centimeter scale, were randomly sampled within each area. This is showed in figure 1 at the bottom right, where you can see the differences between the sectors, areas, and sites. So the ANOVA testing was applied to assess the significance of the differences in the density and the biomass of Branchiostoma caribaeum, considering the 3 random factors, which is the sectors, areas, and sites, with the 5 replicates per area. So the results include variations in the density of biomass, the variability of sediments, variability of contamination gradient markers, and the contamination environmental variables. So table 1 goes with figure 2 to the bottom right, so they go together, And so table 1 is the summary of the nested de novo analysis for the density and biomass. And figure 2 is the mean values of that within the winter and the summer. So table 2 and figure 3 next to that is the summary of nested de novo analysis for sediment parameters, which include grain particle size, carbonate contact, percentage of silt and clay, and TOM, total organic matter. And the figure next to that introduces the mean values of those specific areas. So the use of spatially nested sampling design highlighted the importance of a multi-scale approach to investigate variability in density and biomass. The density and biomass values were consistently lower in the most contaminated areas, which is the inner part of the channel. So we reject the hypothesis that sector scale represents major variability within density and biomass. They found that European landslips became very rare within the northeastern Atlantic coast and the Mediterranean due to human impacts and pollution. Table 3 is the correlations analysis between the biological contamination environment variables, and figure 4 is to the bottom left, which is the mean value and ratios for steroid concentrations along Caatinga Channel in winter 2008 and summer 2009. And figure 5, which is to the right, is the diagram of PCA analysis in August 2008 and March 2009. So we conclude that our results emphasize the need of incorporating sampling designs that account for multiple scales for the evaluation of distribution patterns of like benthic indicators, because these like gradients usually like interact with gradients of natural background variability. The use of a multi-scale approach allows for a better recognition of the process responsible for like the variation patterns. Within this case, what you found the distribution patterns of Branchiostoma caribaeum, variability within the density and the biomass, and you learn that the multi-scale approach allows us to better see variation patterns. So that is all. Thank you.